My name is Claire Whitaker, and I'd like to tell you about a problem that I'm trying to solve. First off, I've got a question. Have you ever bought produce and had it go bad? Did it have that fuzzy gray mold on it? That mold was likely a fungus called Botrytis Cinerea. I'm a graduate student at UC Riverside, and I'm studying botrytis for my PhD dissertation. Now, besides your produce going bad, botrytis causes other problems as well. It can cause food to spoil before it gets to the grocery store and can also ruin other agricultural products like flowers. Due to this, botrytis causes over $1 billion in costs every year. And that $1 billion price tag, it's only for trying to protect crops, not for losses. So how does $1 billion get spent to try and control botrytis? Well, farmers try and use available fungicides, but they don't work well. Then they try using even more fungicides, but it still doesn't kill all the fungus. This is due to the fact that botrytis is very good at resisting fungicides, which were developed with different types of fungi in mind. See, botrytis is a necrotroph, which means it gets its nutrients from dead tissue, unlike other fungi which feed off living plant cells. But botrytis doesn't sit around and wait for plants to die, it actively kills plant cells. This means that its biology is different from other fungi and explains part of why fungicides are less effective. Now, the issues with fungicides came from the fact that scientists originally thought botrytis was not that big of a threat. They thought it was able to release a protein or two which would break open plant cells, letting it slurp up the goodness inside. However, it turns out it's a little bit more complicated than that. Beginning in 2016, people began to re-look at botrytis due to all the damage it was causing. It turns out that there are hundreds of proteins found in the botrytis genome which may contribute to its ability to infect and kill plant cells. These proteins are called effector proteins because they cause an effect on the plant cells. Most of these effectors are released through a known secretion system, but this mechanism requires a tag on each protein marking it for release. But not all the effector proteins have this tag, so how are they getting to the plant cells? My research is examining these tricky effector proteins, trying to figure out how they're moving. If I can figure out how the tricky effectors are getting to the plant cells, it could open up an opportunity to develop new fungicides that defend against botrytis. I'd like to thank all the sponsors of this competition, and I hope you have a great day.